We have very short experience in genomic-assisted breeding. So here is mainly try to report our activities and the status, and also share some thoughts to trigger the discussion later. So in cassava breeding, the recurrent selection has been widely used for more than 50 years, and we have a lot of very good variety released from this breeding method. And the genome-wide prediction has been successfully used in the NextGen project too, especially in Africa breeding programs. And however, for the hybrid breeding in cassava, there has been discussion for more than 20 years, but there is very little effort has been invested in hybrid breeding. So we are going to share some thoughts about that. So that's mainly from the discussion with excellence in breeding, and also with the cassava breeders and CIAT. So before jumping into the before jumping into the topics, I would like to introduce the CIAT cassava program a little bit. So CIAT is a CGIAR research center located in Colombia, in Latin America, and so that region is the origin of the syngene for cassava. So at CIAT we have the largest gene bank, so that's really a valuable source for the global CIAT for the global cassava community. So in the past 50 years, the cassava program have been working closely with the gene bank to identify the different traits. So just several examples: the CBSD high CBSD resistant accessions identified by Dr. Steve Winter in the NextGen project from the gene bank, and also we heard a presentation about the whitefly resistance. And in the Harvard Plus project, we identified accessions with high provitamin A. Let me find the point. So, and also in the RTP food project, we identified the accession with very good queen quality. So not to mention the waxy starch and also small granules. So recently, we started a new project led by CIAT to explore the accessions and also alleles with drought and heat tolerance from the gene bank, and then to integrate the tolerance to the elite breeding program. So and also now for the gene bank accessions, so we have more and more accessions has the genome sequencing data available, so we can do we can analyze the genome sequence for the candidate genes, for example, for the high amylose that's also known as resistant starch, and also for the herbicide resistance genes, and then we can find the new traits and being implemented in the breeding program. So in the past 50 years, CIAT cassava program has developed a close partnership with IIT and also national program. So we have been sharing the treated donors to the improved breeding population to the partners in different regions. And in the NextGen project, and also with the support from excellence in breeding, so we have been doing breeding program modernization following the breeder's equation. So here I will be mainly talk about the hybrid breeding and the genome-wide prediction. So in these slides, I showed the conventional breeding on the left side, and the genome-wide prediction is on the right side. And so our genome-wide prediction strategy is slightly different from what our colleagues in Africa used. So the main difference is just that the training population here is selected from the breeding population. So since we have a small training population, so we do quick clonal propagation in the greenhouse, and then within 2 years we can have the yield trial data and also the genotype data to developed the genome-wide prediction model. And for the breeding population, we really rely on the field evaluation to trim down the population size to about 1,500. Then we can afford the genotyping cost, and to bring the genotype data to the model to predict the breeding value for cycling back and the progenitor, and also to predict the phylogenetic value for variety development. So the close relationship, the training population and the breeding population, they are kind of siblings. So we expect we can get higher predictability, and also compare with conventional breeding. So we know that the genome-wide prediction can shorten the breeding cycle, and not only in population improvement, but also in the variety development. It's about 1 year for variety development. So we started in 2019, and now we are in this stage. So we are collecting the yield trial data from the field and also genotype data. So late this year, we will have some results to show how the cross-validation results look like. And we know the recurrent selection works, and also the genome-wide prediction-based recurrent selection have been successfully used. There are lots of successful stories. So here I will just like to mention one in Southeast Asia. So the variety is called Q50. It's very popular even now. So here is the yield data from FAO stat in Thailand. You can see the yield trend. So the Q50 was released in 1992, and combine the new variety and also the new management, the farmer really get higher yield in their field. But however, in the past 10 years, even the breeding program in Thailand have been releasing new variety, the yield can reach the plateau. Especially now, we have the CMD in Southeast Asia, so we really need to develop a new variety with the CMD resistance and also higher or even better performance and Q50. So the first report of CMD in Southeast Asia is 2015, and now we are still in the development stage on the new variety. So this is really this kind of challenging or difficulty is really trigger us to really thinking seriously about the hybrid cassava breeding. So and so there are lots of reasons we think we need to pursue hybrid cassava breeding. So here I just would like to mention the two justifications. So one is we are really in a changing world, so we must make changes quickly. So especially with climate change, and also we know the CBSD is spreading in Africa, and also CMD is spreading in Southeast Asia. So the farmers in Africa really want to have some upgraded TME 419 with CBSD resistance, and farmers in Africa really want some upgraded version of Q50 with CMD resistance. But however, with our current recurrent selection, we always use some heterozygous progenitors. So every time when we made the crosses, we kind of messed up everything, and so we are going to start from almost from zero, and we have to screen 100 or 1,000 clones and evaluate them in multiple years to find the best one. Hopefully, they can have the target trace and also good performance. But if we have inbred progenitor, the story will be different. So within 2 or 3 years, we can do a quick backcrossing and do trait integration. So we can do targeted improvement and to modify the current variety, or to upgrade the current popular variety, and then deliver the upgraded version to farmers. And so now under the one CGIAR, we really pursue the quality and service excellence. So think about product quality and also the service quality. So in the product quality, there are eight dimensions. I would like to draw your attention to the serviceability. So that means if the product have some flaw or some problem, so we can make quick repairment and then return the product to the customer. So this is something we are not able to do now using our current breeding system. So another about the service quality, would like to see the reliability and responsiveness. So after we receive the request from farmers, we need to provide the variety quickly, maybe within three or 4 years, but under using the heterozygous progenitor and also the current recurrent selection. So it's very challenging for us to deliver the product to provide the service within certain time. So another justification about hybrid cassava breeding is pretty clear. We know heterosis has been widely used in different crops. We believe heterosis will help boost the cassava yield and to help to feed the increased population, especially in Africa. So we know hybrid breeding has been very successful in maize or in corn. So if we look at maize and corn and cassava, we can see both are quite similar. Both are diploid and cross-pollinated, self-compatible, and also show severe inbreeding depression. And both crops are the #1 and #2 starch crop globally. For cassava, there is one advantage is clonal propagation, so we don't need the hybrid seed production system. So this is kind of a dream for the seed crop, like in hybrid rice. They really want to develop the clonal propagation system for hybrid in rice. And I want to show you some yield trend in the major cassava production countries. So here is Brazil in Latin America, and Nigeria in Africa, and Thailand in Southeast Asia. So the orange is cassava and the blue is maize. So on the y-axis is the dry matter yield. So here for cassava, I use 30% of the dry matter consent to calculate the dry matter yield. So we can see basically for cassava, the yield is kind of flat. And this is the national data is maybe the average in each country, and the only exception is in Thailand with the new variety and the new management, real farmer get higher yield. If we look at the corn yield in Brazil and in Thailand, we can see the dramatic yield increase in both countries. At the beginning, in this figure, for example, in 1960, we can see in tropics. So these three countries are all in the tropics, and the cassava really show a very large yield advantage. And so if we even compare with between cassava with corn in the United States, in early stage, cassava have similar yield, but after 60 years of improvement, corn really improved a lot, but cassava still far more behind. We believe there's still a lot of potential for cassava in terms of yield. So
Xiaofei, we have about 2 minutes left.
Oh, sorry. All right, so I will thank you very much. I will be quick. So to think about hybrid breeding, so the first question we need to answer is which product we are going to work with. So after analyze the product profile for the 5 products globally, so we decided to work on the cassava for starch, so which have the basic product profile. Once we develop the system, we should be able to integrate those traits easily and to deliver the product. So work with excellence in breeding and also cassava breeders and CIAT, we developed the system or the process. So so there are several swim lanes. So first we are going to we have aggressive selfing stage, and after we get the S3, we are going to cross S3 to generate the hybrid, and then we are going to genotype and phenotype the S1s, try to understand the inbreeding depression, and we will do test cross, and based on the test cross result to try to determine the heterotic pools. And after we have the pool, so we are going to do reciprocal recurrent selection, start from S1 to do population improvement. All right, so so based on this design, within 5 years, we are going to understand inbreeding depression, heterozygous groups, and also try to optimize the hybriding scheme, develop some variety candidates. I think that's all. Finally, we are looking for scientists to join us to work on genomic-assisted breeding and feature disease discovery and hybrid breeding. If you are interested, please send me email for more details. Thank you very much.